Hello, and welcome to this CADASIL Care webcast. This series is hosted by CureCADASIL, a nonprofit 501c3 organization dedicated to finding a cure for this rare genetic disease. We will be talking to various experts in the field of CADASIL, bringing up topics of interest from patients and families. My name is Sandra Talbert, and I'm a board member of CureCADASIL. My mother was diagnosed with CADASIL and died in 2013, and many of my family members have now been diagnosed. I've been a CADASIL patient advocate ever since. Today, I am pleased to introduce Dr. Beth Kozel from the NIH, working on the ongoing CADASIL disease discovery study in conjunction with Dr. Manfred Boehm, who presented his research in one of our CADASIL Connection webinars in October of 2017. Many of you who are part of this study by the National Heart, Lung, and Blood Institute, or NHLBI, may have met Dr. Kozel during one of your annual trips for this 3-year CADASIL study. Dr. Beth Kozel graduated from Washington University in St. Louis with a bachelor's degree in biochemistry in 1996. She attained a medical degree and doctorate in cell biology and physiology at the Washington University School of Medicine in 2004 through a combined MD-PhD program. Dr. Kozel completed her residency in pediatrics at St. Louis Children's Hospital and her residency in clinical genetics at Washington University School of Medicine. Prior to joining the National Heart, Lung, and Blood Institute at the NIH, she was an assistant professor of pediatrics in genetics and genomic medicine at Washington University. In 2015, she received the Williams Syndrome Association Heart to Heart Award. Dr. Kozel has authored or co-authored more than 30 publications and 3 book chapters and is a member of several professional societies, including the American Society of Matrix Biology and the North American Vascular Biology Organization. Thank you, Dr. Kozel, for your time today to discuss CADASIL with us.
Thank you, Sandra, for that nice introduction. As you said, I'm a clinical geneticist who does research at the NIH. My research focuses largely on rare vascular conditions, including CADASIL. And in my work with those of you who came to the NIH as part of that project, it was unfortunate to learn how few of you had received genetic counseling as part of your diagnosis and journey. And so we wanted to spend some time going through how gene changes impact health for families to help with that discussion. So as this audience knows well, CADASIL stands for Cerebral Autosomal Dominant Arteriopathy, Subcortical Infarct Leukoencephalopathy. That's a mouthful, but the part that we're going to focus on today is the autosomal dominant part. And what that means is that changes in genes, in particular the NOTCH3 gene, can be passed through families, leading to health changes for multiple members of the same family. Before we get into genes, we're going to first focus on chromosomes. So what you can see here is an example of a test that we do in the cytogenetic laboratory that looks at chromosomes. So chromosomes are the vehicles that carry all of our genes. All of us, or at least most of us, have 23 sets of chromosomes, each paired, where we get one chromosome copy from our mom and one chromosome copy of the same pair from our dad. As you can see, we have the 22 pairs outlined here, and then we have a set on the end here that is different depending on if you are a male or a female. Females have 2 of the X chromosomes, and males have 1 copy of X and 1 copy of Y. Each one of these chromosomes carries multiple genes all the way up and down them, and the NOTCH3 gene that causes CADASIL is carried on chromosome number 19. As you can see from looking at this, just looking at chromosomes under the microscope wouldn't be enough to tell us about the NOTCH3 status. The gene itself is just much too small to be seen with the human eye. Consequently, we have to use other tests to tell us about genes. So I like to liken that first test that we talked about, the karyotype, to a shelf of encyclopedias. The karyotype test allows us to look at that shelf and to ask, Is letter A there? Is letter B there? So is chromosome 1 there? Is chromosome 2 there? It doesn't tell us anything about what's inside that book. And what we really need to find out when we're trying to find out if there are problems with the NOTCH3 gene are the spelling of the words that make up that gene. Consequently, in order to understand how the NOTCH3 gene works, we need to do a different type of test, a sequencing test, in order to find whether there are changes that contribute to stroke findings in a family. So genes, unlike our alphabet, are made up of only 4 letters, A, C, G, and T, but they're put together in a very precise organizational pattern that spell out the sort of building blocks of genes and then proteins. Sometimes both parents have a normal spelling of the Notch gene, but when an individual egg or sperm is made, a basically misspelling occurs. And you can see that depicted here as this C being changed to an A. That occurs rarely in individuals with CADASIL. In most cases, a child receives a gene change from an affected parent. And in that way, it is passed through generations. On this slide here, you can see an example of autosomal dominant inheritance. So in this case, we have an affected father who has a capital A or a gene change on one of his chromosomes, while the other chromosome is unaffected. Likewise, mom does not have CADASIL, and both of her chromosomes carry the normal copy of NOTCH3. When each one of them goes on to make either egg or sperm cells, 50% of the time they're going to pass on the chromosome. Dad will pass on the chromosome that has the capital A or the Notch mutation, and 50% of the time he'll pass on his normal chromosome. In mom's case, she has 2 normal chromosomes, so in each of her egg cells, we would expect that she's going to pass on a Notch gene that does not have a gene change. Then, at the time of fertilization, 1 egg and 1 sperm come together, creating a baby. In this case, you're going to end up with 50% of the offspring from this pairing ending up as not having CADASIL, and 50% of the time, or each time that big A gets passed along, the baby does inherit CADASIL. Now, this is depicted as sort of 2 and 2, and that is more of a sort of statistical situation, but it is not necessarily what's true in each family. So in each time, this is sort of like a flip of a coin as to whether the big A or the little a is passed on. In some situations, as you know from flipping a coin, you can get heads over and over again, potentially passing on big A, big A, big A, and so having 3 affected children in a row. Alternatively, you could flip a coin and get tails each time, passing on little a in each situation and having multiple unaffected children. Consequently, having a first child that's affected or unaffected doesn't change anything about the risk to the other, to the next children in that family of an affected individual. Each time that risk is 50-50, independent of what's happened before or after. So when we look at someone who comes to see us in genetics clinic and wants to have additional information about risk to other members of the family, it's not uncommon for us to construct a pedigree. And in this situation, we'll say that it's this person here with the green arrow that's come to see us in clinic. This person is a female, which is depicted as a circle, and she has symptoms of CADASIL, has probably had an MRI that is suggested, and her neurologist has suggested this diagnosis for her. And we do a genetic test for her, and if a gene change comes back that's suggested of CADASIL, she would be given the diagnosis of CADASIL, and we would then talk to her about risk to her children. So because she has the gene change, each one of the children that she has would be at 50% chance of having received that gene change. And so we would suggest that Bob, Jim, Tim, and Ann, when they're old enough, might want to consider genetic testing for CADASIL. Likewise, when we talk to her about her family history, she's likely to tell us that her dad, in this case, also had symptoms of CADASIL, as did many of his family members. As you can see, some of those people have already passed away, as has been denoted by the line through the box here. But some individuals are still alive. And we would talk to her about the likelihood of, given their shared symptoms, that her father also had the gene change. Other people in the family who were similarly affected would have children who would also be at risk for CADASIL. Again, because her father is affected, that means that her siblings also had a 50% chance of having received the gene. So we would talk to her and tell her that her older brothers and younger siblings might also want to consider testing. In this case, we've drawn that her brother, older brother, depicted here, age 47, has also had symptoms. He would be likely to have a gene change, but these other individuals, age 52, 42, and 41, could still be at risk. It's possible that they have the gene change and just have not yet developed symptoms, or that they won't have had the gene change. And until we would do a gene test, we wouldn't know the answer to that question and to be able to tell them whether their children are indeed at risk. So as you can see, risk is highly individualized, and sitting down and talking with a geneticist or a genetic counselor can help make your particular family situation more clear in terms of who should consider testing and who wouldn't be at risk based on other testing that's been done with other members of the family. So going from genes to proteins, how does a change within the CADASIL gene or within the NOTCH3 gene cause a problem for the protein and for how blood vessels work? To a certain extent, that's still something that's up for debate. But what we do know is that when the DNA changes, most of the time, the DNA changes the DNA changes in a very particular way. So the base pairs that are laid out in the DNA are laid out in sort of sets of 3, and those sets of 3 go on to code for a specific part of the protein called an amino acid, which are the building blocks of a protein. And the amino acid that's most commonly affected in CADASIL is a cysteine. Cysteines are very important amino acids in the building of proteins because they have this side chain here that includes this S, which is a sulfur molecule. And when two sulfur molecules get close together, they can get linked together and cause portions of the protein which might actually be distant from each other to be held closely together in terms of the 3D shape of the protein. Consequently, if a gene change happens such that a cysteine is no longer formed because the changeover from those base pairs to the protein doesn't occur right, this bonding doesn't occur properly, and the expectation is that the resultant NOTCH3 protein would be somehow abnormal in that because it doesn't have the right 3D shape, it doesn't function in the same way. And so that's why we think that the majority of gene changes are affecting cysteine, is because they're affecting those important shape of the NOTCH3 protein. Interestingly, if you've had a gene test, you probably have looked at the report, and on that report, it gives you a very specific set of numbers where it says there's a gene change in this place. It actually probably gives you 2 numbers. One is the base pair number, and one is the protein number. And when you look at those numbers, if you take a whole large group of people who have CADASIL and you look at the places in their gene or protein where they tend to have problems, the bulk of them lump in this front half of the molecule. So within these first 4 in particular, but up to 6 exons is where the sort of majority of people who have features of CADASIL have gene changes. There are people who have gene changes further back in the molecule, but the majority of them are up front. Interestingly, back in the day, which would be actually only not too long ago, we only did genetic testing in situations where someone had features of a particular condition. And when they had, in this case, features of CADASIL, we would sequence them. And we found exactly the pattern that I just showed you on the last slide. But more recently, with advances in technology, we've had the ability to sequence large numbers of people and look, instead of just at their notch gene, to look at all of the genes across their exome. And we started doing this for people who had CADASIL features, but also for people that we believed to be entirely healthy. So we just took anybody who was interested in donating their DNA to these types of procedures. And the data shown here is from another group who published an article that's listed at the bottom in 2016, where they started to look at the frequency of gene changes within the NOTCH3 gene in patients in general, so not necessarily those that had features of CADASIL. And what they found was, while we believed previously that somewhere between 2 and 5 people in 100,000 people had the disease CADASIL, we were actually seeing gene changes much more commonly. So in the South Asian population and in the East Asian population, they were as high as almost 10 out of 1,000 people had a change within the NOTCH gene. And that suggested, or the NOTCH3 gene. So that suggested that just having a gene change doesn't necessarily give you the features of CADASIL, that it might be important where that gene change was within the molecule. And so in this study, they took a group of people who had features of CADASIL, and these pink lines above show basically the frequency of gene changes in that part of the molecule. And like what I showed you earlier, for the majority of patients who had features of CADASIL, their gene changes are in the front half of the molecule, the front piece. But when they took people at random, so people who didn't necessarily have features of CADASIL, and sequenced them, the gene changes they found affecting cysteine molecules as well tended to be in the back part of the molecule, or at least not focused as much in the front. And so that suggested that, again, that having a gene change itself in CADASIL may lead to CADASIL features, especially if it's in the front part of the molecule, but we were a little bit less certain about the gene changes that happened in the back end. So in this study, they looked at both the brain volumes and the amount of strokes in people who had gene changes in that front part, and showed that there were significantly more features in terms of volume loss and numbers of infarcts in people who had those front of the molecule gene changes, as opposed to people in the back who had gene changes in the back. Now what you can see is that people who had gene changes in the back of the molecule are not at zero. They do still have changes with volume loss and number of infarcts, but it seems to be less. And so that suggests that some of these gene changes may lead to more severe phenotypes, while other changes, as in those in the back, may lead to more mild phenotypes in terms of features of CADASIL coming on significantly later in life. And for some patients, that may mean that they actually don't have features of CADASIL, or at least not that any have been detected clinically at the time that they pass away from potentially other conditions. And so that means when we do genetic counseling, we want to pay attention not only to the fact that someone has a gene change, but where it is in the molecule and what it means for that individual. To a certain extent, that's something that we're still continuing to learn. And in the webinar that we have on understanding genetic testing later on, we'll talk a little bit about those differences in variants and how those will be reported to you in terms of understanding the likelihood that you will have symptoms from that. So understanding penetrance and expressivity. Having a gene change in NOTCH3 puts you at risk of having features of CADASIL. Most people, if they live long enough, who have gene changes within this gene, especially in the front part of the gene, will go on to develop features of CADASIL. However, when that occurs and the degree to which that occurs can vary. Some of that, like we talked about, has to do with where that gene change is, front of the molecule versus back of the molecule. But even within the same family, where people are all carrying the same misspelling, the same gene change, there may be variability. As a result, there are likely modifiers, things in the environment that impact when someone will have gene changes, will have features of the condition. There may be other genes as well in the genetic background that influence the severity of the disease. Those are all still things that we're looking into so that we can begin to give better sort of personalized expectations for an individual based on their gene change. But it's important to note that even within a family, the onset of symptoms and the severity of those symptoms may be different. So in summary, gene changes in NOTCH3 cause CADASIL. Most of these changes are inherited, though a very small number are new in the affected person. These changes, once in an individual, can be passed from parent to child on chromosomes. For each pregnancy to an affected person, there's a 50% chance of passing the gene on. So that means if I have CADASIL, every time I get pregnant, 50% of the time that child will inherit the NOTCH3 gene change. Most of the gene variations that cause CADASIL affect those cysteine amino acids. However, the severity of the disease features likely varies based on where the gene change is within the gene and some additional environmental modifiers that are yet to be discovered. We need a whole lot more research to understand how all of these things work together to cause disease. And to work towards a cure. And we're grateful for those of you who have worked with us in the past on our study and hope to continue to work towards a future cure coming soon.
Great. Thank you, Dr. Kozel, for this helpful presentation and making the genetics of CADASIL, which can be confusing, a bit easier to understand. We do have a couple of questions for you from our community, if you don't mind.
No problem.
Great. Okay, so the first one was, I was diagnosed with CADASIL, but both of my parents are fine. Does that mean I have a de novo mutation, or what does that mean?
That's a good question, and it's a question that we hear a lot. So a lot of that, again, like we talked about with the pedigree, depends on the family. And so if you were to have genetic counseling, we would sit down and go through all of the situation for your family. But the short answer is we can't say, based on the information that you provided, whether your variant is de novo or inherited. Statistically speaking, it is most likely that it is inherited and that your parent just has not yet shown symptoms of the condition. And we would recommend that at that point that if they were interested, that they consider testing for themselves. De novo gene changes do occur, but they are much less likely than an inherited form.
Okay, thank you. A second question is, if I have CADASIL, does that mean all of my brothers or sisters could have CADASIL? And we received a similar question from a parent that said, does having CADASIL mean that all of my children could get CADASIL?
So, as we talked about, if a parent has a gene change that causes CADASIL, every time they make an egg or sperm, there's a 50% chance of that particular egg or sperm receiving the affected gene. And so every time an egg or sperm is passed on, it's a 50-50 chance. Some people, unfortunately, could be unlucky and have multiple or even all of their children affected with CADASIL. Likewise, some people could have a situation where they, by chance, pass on all non-changed or non-mutated genes, and all of their children not have CADASIL, or any combination in between. We just really can't predict. At each case, it's 50-50. Now, I can say that if you listen to our second webinar, we will talk a little bit about prenatal genetic testing and preconceptual genetic testing that some people who are affected with CADASIL might want to think about or know about before they decide to have children of their own. That will impact that. So do come back to listen to our next webinar on that subject for additional information.
Great, thank you so much. We appreciate your presentation and all of the explanations for the many patients and families with questions. CADASIL Care is made possible by volunteers of cureCADASIL, our speakers, generous donations from CADASIL families, and a grant from Global Genes. This concludes our presentation. Thank you for joining us.